Hello and welcome to Nikolay's genetics lessons. Today I got a question from one of my subscribers, and here's the problem. What is the maximum number of isozymes that can exist for heterodimeric protein that consists of combinations of 4 different polypeptides? So here is different variants of the answers. As usual, I recommend you to pause video here, try to solve this problem on your own first, and when you would be ready, you can run video again and you can compare your answer with my answer and explanation. So, let's first specify what each term means. For example, isozymes consist of 2 parts, iso and zymes. Enzymes, as you probably already guessed, stands for enzyme. Iso meaning that these enzymes perform the same function but result of the different genes. So these enzymes are not in the same are not alleles of the same gene. And just compare in a contrast other enzymes. Other enzymes would be just alleles of the same gene. So would be basically the same polypeptide chain, but that would have different mutations, and due to these different mutations, these other enzymes would perform the same function, but probably with different effectiveness. So it is very important for our organism to have different enzymes or isoenzymes that would perform the same catalytic reaction, but we would use them, our organism would use them as fine tuning of these reactions. So let's now talk about what heterodimeric protein means. So hetero and dimeric. Also, this word consists of 2 parts. So hetero means the same as, for example, it's maybe would be easier for you, heterosexual. For example, I'm heterosexual. I have a wife and 3 beautiful children. But there are people who is homosexual, meaning man with man or woman with woman. They don't have beautiful children. So hetero means different, 2 different, like man and woman. And dimeric means D stands here for 2 and protein. So this is protein that consists of 2 subunits that are different. So now we are ready to solve this problem. And also I want to bring to your attention that isozymes and allozymes usually those meaning different things, but these 2 terms used interchangeably. So sometimes you have to find out what this or that scientist means when he is using isozymes, whether it is isozymes that result of different genes, structurally different proteins that perform the same function, or whether the scientist means allozymes, that is just different alleles of the same gene. So we also know that our heterodimeric protein consists of combination of 4 different polypeptides. So 4 different polypeptides. Let's make a small table. So I would use different colors to designate different subunits of this heterodimeric protein. So one color could be white. Let's it be global protein, and this is going to be another variant, and this is going to be the 3rd variant of the subunit, and the green color for the 4th variant of this subunit. And we can make a table. So on other side we would use the same colors So this is going to be another subunit, second variant of subunit, and third variant of subunit here, and the last one would be a green one. So now when we build a table, we can find all possible combinations that these 4 subunits can make in a heterodimeric protein. So we would have 4 columns here, and we'll have 4 rows. But do not assume that if we have 16 cells here, we are going to get answer D16. It is not so. I will show you in a minute. So here we would have heterodimeric proteins that consist of 2 these subunits, that is the same. Here we would have in the next cell 1 subunit blue, another subunit white, and in the next row we would have red subunit and white subunit, and in the last we would have green subunit and white subunit. So in the next column we would have white subunit and blue subunit. Then we would have blue subunit and blue subunit. Then we would have red subunit and blue subunit, and then green subunit and blue subunit, white subunit and red subunit here, blue subunit and red subunit here, and red subunit and red subunit here, and in the last we would have green subunit and red subunit here. So the last column, wait just 1 more minute, so white subunit and green subunit here, then blue subunit and green subunit, and then we have red subunit and green subunit, and we have green subunit and green subunit here. So now I want you to pay attention that we are looking for heterodimeric protein. Hetero means 2 different, but as you see, this is 2 of the same, this is also 2 of the same, this is 2 of the same, so we can cross out this variance. So now if you think that we cross out 4 variants and the answer would be C12, you're also not right, because I want you to pay attention here that, for example, this variant and this variant are the same. If you flip this protein over the vertical axis, you would get the same protein as it here. So it's not left and right form of the same heterodimeric protein, it is the same protein. We just turned it over the vertical axis. But I also want to tell you that all these proteins, the rest proteins, have 2 variants here. So red and blue, and red and blue here, and for example green and white here, green and blue here, and green and blue here, and green and red here, and green and red here. So we can cross out all the duplications, So now when we get rid of all duplications, let's count how many unique variants we have. 1, 2, 3, 4, 5, 6. 6 variants. So as you see, the correct answer is answer A. And this is all for today. Thank you for your attention. Please subscribe for my new videos that I post almost every day. Thumbs up if you like this video. Please write your comments, questions if you have any. And see you in the next video. Goodbye.